Okay, hi everyone. Thanks for those of you who did make it in person today, and hi everyone else on Zoom. So today we're joined by David Benkeser, who received his bachelor's and master's in biostats from the University of Georgia and his PhD in biostatistics from the University of Washington before doing a postdoc at UC Berkeley and then joining us here as faculty at Emory in the biostatistics department. David is an associate professor and primarily leads methodological research with a focus on the theory and applications of machine learning in causal inference. Specific areas of interest include vaccine statistics, computing risk, complex longitudinal data, and theory of robust non-parametric statistical inference. And he has done work on preventative vaccines, tuberculosis, and HIV prevention. He also teaches a data science class, and his favorite bakery in Atlanta is Alon's, I think. Today, he will be telling us about vaccine correlates of production across scales. Welcome, David.
Yeah, I know. Maria's really put me in a tough spot here. Number one, it's got to be a little tart. If you haven't been to a little tart bakery, fantastic. As long as it's fine, it's on my way to work. That's probably what I get. Anyway, nice to be here. Thank you for the invitation. Nice to see you all. People online, I don't know where the camera is. Hello. Yeah, so as Maria mentioned, I'm a faculty member in the Department of Biostatistics and Bioinformatics here. And I know you'll all be very disappointed to hear that I'm not really going to be talking a lot about biostatistics and bioinformatics today. Lance, I'm sorry to disappoint. Please don't tell the others in our building. Yeah, I wanted to give just a very high-level overview of a very, very large area of research that I've spent a lot of time in particular since the COVID pandemic. And that area is vaccine correlates of protection. And I guess I tried to make it sort of more relevant to the particular room we find ourselves in by trying to emphasize a little bit that really I think this is an area of research in infectious disease that does span really a lot of different scales. So we'll talk a little bit about, how understanding things at a very fine-grained biological level has implications then for vaccine development, for product development, for product deployment, and really impacting human life. So hopefully I'll give you a little bit of overview here. I'm going to talk probably a little bit out of my comfort zone a bit here because this is kind of more general science than statistics. Luckily, Cheryl is here, and I'm going to look at her every time I say anything about lab science, and you make some face when I start saying things that are wrong, and I'll really appreciate that. Okay, so here we go. Okay, so I start with a friendly reminder because, boy, it's a weird world that we live in today. And I think this message, is one that we can all agree on, but also one that we can remind ourselves to promote in spaces that we're able to. The development of safe and effective vaccines is one of the, if not the greatest public health achievement ever in history, full stop. So again, I think this is a safe space to say that. I have tenure now. I guess this is going on YouTube. They can't get me, right, Lance?
You're safe.
We're safe here. Okay, but again, this is just a message that I think, we can continue to promote in the spaces that we can, whether that's with friends and family or strangers in a bar, wherever we can get this message out, I think is a good thing. Okay, so what about vaccines? How do they work? How do we think they work? And I put "we think" there because that's kind of part of the talk today is how do we actually learn about how vaccines work? I think we have a very good understanding of base biology and what we think is happening when we give you an injection, for example, to deliver a vaccine, right? But how do we start to develop kind of an empirical body of evidence that supports this kind of picture? So let's start with the picture, get everybody on the same page about the ways that we think that vaccines are working to protect us from infections and diseases. And again, there's probably people in the audience that know a whole lot more about this than I do. This is my crude biostatistical understanding of vaccine biology. So we make a vaccine. There's lots of different ways to make a vaccine. And what I'm showing here is a nice little diagram from, I think, the Washington Post put this out during the COVID pandemic to explain how vaccines work. This particular picture is talking about vaccines that are made using what we call virus-like particles. So those are, particles that look like viruses, but they don't have any genetic material. So when they enter your bloodstream, they're not going to replicate and create new viruses. Or on the bottom there, we maybe make vaccines that we call protein subunit vaccines. So we can grow little proteins in a lab inside grasshoppers or in eggs or whatever magic they're able to do in labs. I don't know what Cheryl does over there, but they can grow proteins that look like a little piece of a virus, for example. So here, the SARS-CoV-2, you've probably heard about the spike proteins. That's what is going into the protein subunit vaccines. In any case, these are little blips of a virus that we're going to present to your immune system in some way. So for COVID vaccines, pretty much all injectables now. So you get an injection that triggers some kind of inflammatory response that tells your body, hey, send some immune cells to go and check this out. A key kind of feature of that immune response is those APCs in the middle, antigen presenting cells. They gobble up little bits of those viruses, and then they spit them out, these little viral peptides that they send on to your helper T cells. Your helper T cells then deliver those little viral peptides and information about them onto other immune cells. So T cells, for example, and B cells. And B cells can go on to create little proteins that are called antibodies. And so antibodies are going to play a big role in the discussion today because antibodies play a big role in why vaccines work or why we think vaccines work. So that's down here on the bottom. What these B cells make, it's these little guys that look like Ys. These are these little proteins, and they do a lot of things. One of the things that they're really good at is sticking to things. So they're good at binding to proteins on viruses, and that's what we think a little bit about how vaccines are supposed to be protective, is because you make all of these antibodies, and you have all these little proteins floating around in your blood, such that when a virus is introduced, let's say, to your bloodstream, those antibodies stick to it and stop that virus from doing what it wants to do. And what it wants to do is infect your cells and make you sick. Okay, so this is a diagram, kind of showing what we think based on basic science about how vaccines work. Okay, but how do we know that vaccine? I mean, this is all well and good, very interesting biologically, but on the other hand, like from a clinical perspective, who cares, right? We just want to know that if I give you a vaccine, I'm reducing your risk of infection or disease. So how do we know that actually vaccines are working? And we know that through a very careful and pragmatic approach to vaccine development. So this is, again, I think something most people are familiar with, but if you're not familiar with like sort of drug or vaccine product development pipelines, I want to just highlight this here because we're going to come back to both of these slides and kind of point to where vaccine correlates of protection play a role across this whole sort of lifespan of product development, and as well as across some of these different immune pathways on the previous slide. Okay, so how do we know the vaccines work? Well, this is the whole pipeline. We start to know that vaccines really work once we get over here in these phase 3 and phase 4 trials, these very large studies, but it starts with something very basic. So we start doing things in the lab, like figuring out just what kind of little particles we should put into our vaccines, what should those proteins be, right? How should we think about presenting to them? Should we add like adjuvants to the vaccine to increase your inflammatory response? All of these sort of basic questions about how we should construct a vaccine. We can do studies in animals, right? So like a mouse model or non-human primates, et cetera. So we can start giving these vaccines to animals and seeing if they have some response. Are those animals generating immune responses? If we challenge them, try to make them sick, does the vaccine prevent them from being sick? Once we're pretty confident that those vaccines are doing something, we can start doing trials in humans. So a phase 0 trial, we like give an injection to somebody, we close our eyes, cross our fingers, and kind of hope they don't die. If that's the case, then we'll move on to like a phase 1 trial where we'll get a few more people, give them injections, make sure that they're not getting too sick, having too severe reactions, and we start to sort of build a body of evidence that these vaccines are actually safe. So we start to characterize how reactive people are once they receive these vaccines. We also start to characterize their immune responses, which is again kind of the whole story of today. What are these immune responses and how do we learn about how they work? Once we're pretty confident in phase 1 that our vaccine is doing something in humans and is not going to kill a lot of people, we move on to phase 2, where we start to do slightly larger trials. So maybe now we've selected a dose of the vaccine. We've agreed that this dose is not going to give people like terrible rashes or bad fevers. This is the dose we're going to move forward. In phase 2, we start to do longer term studies of these vaccines. We see not just do you have an immune response, how long does that immune response last, for example? And maybe we start following people up to record some endpoints, like are they actually getting sick or are they actually getting disease based on the pathogen that we're interested in. Once we think we have some promising results from phase 2, we move to phase 3, which is like the big money trial where we enroll 20,000, 30,000 people. We randomize them to receive a vaccine or a placebo or a control vaccine, for example. And now we really check, is this vaccine doing what we actually care about, which is preventing infection and disease? Okay, we do that in the context of this randomized trial. Hopefully we get some positive results. We go to regulatory bodies like the FDA and say, look, our vaccine seems to be preventing infection and disease. Can we please go and sell it for a lot of money now? FDA hopefully says yes, and then we move on to post-licensure studies in phase 4, where that's, where we're studying rarer safety events, we're studying more effectiveness in the real world, and that's the whole vaccine development pipeline. And so where I'm going with this is to talk about the actual topic for today, which is vaccine correlates of protection. And this is about really how we can learn about, through this vaccine development pipeline all along the way, how we can try to learn about this picture, whether we can confirm that this picture is actually what's underlying, the effectiveness that we see in clinical trials and in the real world once we deploy a vaccine. And so we call this kind of area of research trying to establish a vaccine correlative protection. So for now, I'm going to just sort of leave a very high level definition out there, and then I'll tell you later that people have a dozen different words for the same kind of concept. But the high level concept here is that vaccine correlates of protection broadly are some kind of immune response that we can measure that will predict whether a vaccine will be effective at preventing some kind of clinical endpoint. So again, whether that's infection or disease or severe disease or death, right? Some kind of clinical endpoint, and we want to be able to look at an immune response and guess whether that vaccine is going to be good at preventing that endpoint. These are often referred to, I tried to figure out who the first person to refer to this as, but it's like 100 different people have said this in the press and in scientific publications. These are often referred to as the holy grail of vaccine development. If you can establish a vaccine correlate of protection for a vaccine and or a particular disease, it is a huge deal because it enables you to do a lot of very cool things. So I think the next bit is me trying to convince you that understanding not just whether vaccines work, but how they work has a lot of benefits. And those benefits span from early phase vaccine development to late stage decision making about how vaccines are deployed and whether vaccines are updated, for example, to accommodate new strains. So why are correlates of protection so important? Why do we call these the holy grail? So we can start with like early phase development. As I said, when we're doing these sort of like phase 1, early phase 1 trials, phase 2 trials, even in animal studies, right? What we're really looking for very often is some kind of immune response, right? And what we want to understand, use that knowledge to help us with, is selecting, for example, a dose for a vaccine. So what I have on the left there is one of the early publications for the Moderna COVID-19 vaccine that came out, where they were looking at immune responses and, of course, reactogenicity and safety to try to decide on a dose of the Moderna vaccine. And so what's shown there on the left, the vertical axis is some kind of immune response. If you know or care about these things, right, one is a binding IgG response, one is a neutralizing antibody response, and they're showing each line as a participant, and they're showing an individual's immune response over time, okay, following the first dose and the second dose. And you can see that the red line is the higher dose. That's if you squint, you can see that's the 100 microgram dose of the vaccine, and the blue is the 10 microgram dose. And so based on this picture, Moderna decided that those immune responses that you're getting when we give you the red vaccine, right, are worth it. They're worth it, and it's worth having a bad rash on your arm and having a horrible fever for the next 2 days because your immune responses are so much better, right? And so this is the type of information that very early on in vaccine development, already we're starting to look at these immune responses, right, and make decisions based on them. The middle panel is another one. So this is the RTSS malaria vaccine. This is one of 2 now licensed preventive malaria vaccines. This is a paper from quite a while ago, I think in the 90s, where they were looking at different adjuvants to add to a vaccine. So adjuvants are basically what we learned, this is my understanding, what we learned is if we give you just like sort of pure proteins, your body doesn't always react to them. It doesn't even notice that they're kind of there. So if we kind of give you very pure formulations of a vaccine, they might not be as effective as if we add something kind of dirty into the vaccine. That's what an adjuvant is. It's sometimes, so this is my understanding, something that stimulates kind of an inflammatory response that really turns on your immune system and say, hey, there's something going on here. Let's go and check it out. So this picture here for RTSS was a study that was trying to figure out what kind of adjuvant to add to the RTSS vaccine. They settled on an adjuvant called, what is it, AS01E. GlaxoSmithKline makes it, and unfortunately, GlaxoSmithKline has decided to mostly make that adjuvant for their shingles vaccine and not for this vaccine. So that's a whole other fun story that you can Google and get a little bit depressed about vaccine development pipeline. But anyway, in 1998, we were very happy to see that this adjuvant led to better immune responses. You can see some very small p-values there, and that led to some critical decision-making for how that vaccine progressed through the pipeline. On the right side, so another thing we can use very early on to help with is antigen selection. So the right one is showing similar immune responses, a little bit of a different way of presenting them. These are reverse cumulative distribution functions. I can't help myself, I'm a statistician. So it's basically showing for each level of immune response on the horizontal axis, what fraction of the population had an immune response at least that high. And they're showing that for a few things over time. So here they were really trying to say, actually by putting 4 different strains of a particular bacteria into a vaccine, you can get more robust immune responses than others. So maybe we want to move forward with that sort of quadrivalent formulation. So if you can trust, right, that these figures, what's shown on the vertical axis, at least in the left figures, right, is telling you something about whether this vaccine will work or not, right? That's what immune correlates are all about. We need to trust that those immune responses that we're plotting here are telling us something useful about whether these vaccines will work. If you believe that, right, then you can make really critical decisions very early on in the vaccine development pipeline based on just those immune responses, right? We don't have to wait and do these very large scale trials to figure out what adjuvant we should use, what kind of dose we should use. We can just study very small studies, study immunogenicity, these immune responses, and make these critical decisions based only on that alone. This requires a leap of faith, right? You have to believe that these are the right immune responses, that you measured them in the right way, that this is not leading you astray, right? And so that's really the whole story about vaccine correlates of protection. If you can believe that, then you're really in business. Okay, why else are correlates of protection important? So moving a little bit further down the pipeline, how else can we use this information? And this is where I think, maybe most people think about correlates of protection as being potential surrogate endpoints that can be used in later phase studies. So the idea here is that doing a phase 3 clinical trial is extremely costly. So if you think about the COVID-19 vaccine trials, those were all, the ones in the US were all at least 30,000 people. Some were as high as 50,000 people. Followed up for 2 years with study visits very intensely during the first few months and at least every 6 months after that. That costs a lot of money to run. Okay, and why do we need them to be that large? Right, because we need to enroll enough people that we see enough endpoints that we have power to detect if our vaccine is actually working. That we're gonna learn that from the phase 3 trial. Right, so these trials are very costly because we have to enroll a lot of people to be able to see these clinical endpoints, right, that we actually care about. But wouldn't it be nice if instead of having to wait for all these people to get COVID or not, or get severe COVID or be hospitalized with COVID, wouldn't it be nice if we could just look at an immune response, let's say give a couple hundred people a vaccine, look at the immune response and check that the immune response is like good enough. All right, and if we could do that, we could avoid this whole costly phase 3 process. We just say, you know what, we gave these people a vaccine, all of them had this great immune response. We know that if they have that great immune response, this vaccine is going to be effective, so poof, we're done. And this vaccine we know is going to be a good product. So this is something that is what we're really trying to do with correlates of protection a lot of the time, is to be able to make sort of regulatory decisions based on what we would call a surrogate endpoint. So a surrogate endpoint meaning that we're swapping out the clinical endpoint, infection or disease that we actually care about, we're swapping that out for a surrogate, which is our correlate of protection, right? Which is just this immune response that we can measure. And if you can do this, right, then we can speed along this whole pathway for approving vaccines. So the one where we think that, maybe is the canonical example where this has actually been possible and codified by different regulatory agencies is for your seasonal flu vaccine. So I'm showing a little snippet down here from the FDA guidance for industry for seasonal flu vaccines. And the idea there is basically, you can't do randomized trials every flu season. It would be pointless, right? We would be learning whether this flu vaccine works or not at the end of each flu season. So great, we can approve a product, right, that will never see the market because by the time the next flu season comes around, flu has moved on to a new strain and that vaccine no longer works, right? So that sort of traditional phase 1, 2, 3 development pipeline for flu vaccines is a little bit challenging. So regulatory agencies noted this and have therefore created accelerated pathways for getting products approved so that we can get flu vaccines in arms in time for the flu season. So this is a little snippet from that. I'll just point you to this little bit down here that says that we can base an accelerated approval upon some kind of well-controlled clinical trial that establishes that your vaccine has an effect on a surrogate endpoint that is reasonably likely to predict clinical benefit. So again, they're basically saying if you have a correlate of protection, right, and you can convince us that this correlate of protection is well established for your product, then we can give you approval for your vaccine based on that sort of immune response alone. I'll give you another example that is currently very much timely and relevant for this question around surrogate endpoints for vaccines. There's currently an outbreak of Ebola Sudan that's happening in Uganda. right now. And you might remember, the 2014 outbreak of Ebola, the one that, made the news, lasted for a very long time, thousands of cases, many deaths. That was the Ebola Zaire virus, so a different flavor of Ebola, right? Very much related, still Ebola, right? But two flavors of the same virus. There is an approved virus, approved vaccine for Ebola Zaire, right? For the one that happened in 2014, they got a trial into the field. They did a phase 3 study. They showed it worked, and it's now licensed and approved. There is no approved vaccine for Ebola Sudan. The vaccines that we have, many of the vaccines that we have in the development pipeline for Ebola Sudan look a lot like the vaccines that we developed for Ebola Zaire. We can show in preclinical data they have the same immune responses. They're pretty much behaving the same, right? But so far, regulatory bodies haven't been comfortable saying that this jump from Ebola Zaire to Ebola Sudan, just because we saw that over here for Zaire, does not mean that it's going to translate for Sudan. So as a result, in this current outbreak of Sudan, WHO is trying to launch a new phase 3 randomized trial to try to establish that this works. I'm not making a comment on whether this is right or wrong. I'm not a filovirus expert. It's just to say that these sort of questions around when is an appropriate body of evidence such that regulatory bodies can make this kind of decision based on immune responses is a really complicated and very timely question for a lot of diseases that are out there. Another way correlates of protection are used is they form the basis for expanding indications of vaccines. So we saw this with COVID, is that very often when we initially study vaccines, special populations are excluded from those phase 3 trials. Again, I'm not here to comment on the ethics of this. Plenty of people have written about that. Should we include children and or pregnant women or immunocompromised individuals in these trials with more regularity? The fact of the matter is, for the most part, vaccine manufacturers are a little bit uncomfortable with starting clinical trials in children. They want to see that it works in adult populations, and then they'll start giving it to younger and younger children. With COVID, for example, the decisions to license vaccines indicate vaccines for kids under 16, under 11, and now eventually under 4 and 5. All of that was based basically on immunogenicity. Of course, there was safety data as well to support this, that these were safe for children. But all of the effectiveness evidence was basically inferred by saying that the types of immune responses we see in kids don't look that different than the types of immune responses we see in adults, right? And so as a result, regulatory bodies were comfortable saying, okay, now we're recommending that kids get COVID vaccines as well. So that's another way that correlates of protection can help us. The other one here, also timely and relevant, is updating strains. So again, sorry to talk so much about COVID, but it's something that's familiar to everybody and something that's still very relevant when we talk about when is it time to get an updated strain in our COVID booster shots. Okay, so this is from recent regulatory guidance as recent as last year, where FDA was basically saying we prefer COVID-19 to have some kind of SARS-CoV-2 virus that looks like this JN1 strain that's like an Omicron subvariant of COVID. And all of that decision is based on saying, hey, the existing vaccines that we have don't give you a good enough immune response to fight off these new variants of COVID, right? So they're not going out and doing new randomized trials. They're not going out and doing effectiveness studies, right? They're doing small immunogenicity studies where they say, basically, we can no longer sort of neutralize this new version of COVID that's out there circulating. So we need to update the vaccine as a response. Okay, so another place, vaccine correlates of protection. Hopefully I've convinced you that at least there's some utility in these, but it's really all over the place here. So far we've hit on, very early on, they're helping us decide how to make our vaccines. A bit later on, they're helping us maybe be way more efficient with our decisions about approvals for vaccines. And then as we move into post-market licensure, they're even helping us decide when our vaccines need to be updated. Another sort of post-market licensure area that is maybe a bit boring, but actually I think pretty critically important, is to support technology transfer and lot-to-lot consistency studies of vaccines. And so the idea here is that there's a big push in the vaccine development community to be able to have countries in the developing world manufacture vaccines in country, right? So that they don't have to go to, rich Western countries and beg for vaccines when they need them. No, they can be manufacturing this on their own. So as a result, there's a lot of effort in sort of taking technology that, maybe is developed in labs in like the UK or the US, taking these vaccine products, right, and now manufacturing them at scale, let's say in India or somewhere in Africa. And in order to do this, you want to be able to, be sure that, yes, we showed that when we make this vaccine, let's say in the UK, it's safe and effective. But now we need to do a study that says the vaccine that we're making in India, right, looks approximately the same. And so another way that we can do that, at least in terms of effectiveness, is to study correlates of protection, to study immune responses, and to make sure that the immune responses generated by these vaccines look about the same, irrespective of where you manufacture them. So there's a sort of boring table on the left. Don't look too hard at it, just showing that we did this for CoronaVac. That's a live attenuated COVID-19 vaccine. There's now a licensed dengue vaccine. Actually, I'm not sure that one's licensed just yet, but it has had some promising phase 3 results come out. And there's, a result of a study of a tech transfer of that to, gosh, I can't remember the country now. But in any case, this is just pointing out that, even after we have vaccines that have been approved, they're being manufactured. If we want to start manufacturing them somewhere else, immune correlates of protection can help support that. Okay, so in 28 minutes, hopefully I've convinced you that correlates of protection, if you can get them right, you can identify the right sort of marker that a vaccine is working, you can do a lot of cool stuff with it all along the vaccine development pipeline. So now we'll get a little bit sort of more into the nitty-gritty of what do we mean when we say we have like the right immune marker, or that we are claiming that some kind of immune measurement that we're making is in fact a correlate of protection. And so to do that, we're going to add like a little bit of sort of substance and a little bit of kind of nuance to our definition of correlates. So this was a field that I would say it's still a field that's quite confusing in terms of nomenclature, because there were several different groups for about a decade that were publishing papers using different words for the same thing. So eventually they got together, and in this paper, and the DOI is down there if you want to read it, they got together and sort of tried to have an agreed upon set of terminology. I don't know that it's entirely standardized yet across the field, but for the purposes of this discussion, this is a nice distinction to sort of make here. Okay, so when we say correlative protection, as I said, basically this is something that we think predicts whether a vaccine works or not, right? So it's some immune marker, some way of measuring an immune marker, right, that is going to be correlated with vaccine efficacy. So that basically, as I said, predicts whether a vaccine will work or not. Now we can further break that down into two subclasses. So correlates of protection can be either mechanistic or they can be non-mechanistic. So mechanistic correlative protection, right, is something that is causally related to the protective efficacy of the vaccine. So it's actually, right, measuring one of these little functions that we have here. It is along this causal pathway, right, that this is actually the reason causally why this vaccine is leading to protection. And that's probably the way that you're thinking about these, based because I showed you that slide at the beginning, right? That you're already probably thinking about most correlates of protection as being mechanistic. This is some mechanism that your immune system has for preventing you from getting infected or sick. But there's also other forms of correlates that may be non-mechanistic. Okay, so these can still be used to predict whether a vaccine will work or not, but they don't necessarily have to be the mechanism of that vaccine working. So I'll give you a very simple silly example of a non-mechanistic correlative protection. So this is, of course, completely hypothetical, but I think it drives home the point. You can imagine that you give a vaccine to some people, and that vaccine induces some kind of inflammatory response. Okay, that inflammatory response goes on to stimulate your immune system to make all these great mechanistic correlates of protection. That's along this pathway at the bottom, and those lead to protection. Okay, but also that inflammatory response makes your hair turn purple. Okay, so purple hair, at least in this population, could be used as a surrogate marker for the vaccine work, right? So if you give this vaccine to a bunch of people and they have purple hair, right, you might convince yourself that actually that's pretty predictive of whether this vaccine will work, right? Because it induced this inflammatory response that I know induces these immune responses that lead to protection. But obviously having purple hair is not mechanistically related to whether you're protected at all. It's not on the causal pathway, right? But it is something that could potentially reliably be able to predict whether or not this vaccine works. And so that's a little bit of a silly example, but it's one that's not so far-fetched when you start to think about the ways that we measure correlates of protection. So maybe a slightly more realistic example would be something like measuring antibodies that are circulating in your bloodstream when we're talking about a respiratory infection. Right, so when you have a respiratory infection, where are you being exposed? You're being exposed through your nasal passages, right, and in your lungs. It's pretty hard. I think we can do it, but it's very invasive to measure immune responses in your mucosa, right, in the linings of your throat and of your lungs. right? So we don't typically do that. What we do instead is we take a little blood draw and we see, do you have antibodies in your blood, right? And if you have antibodies in your blood, the idea is you probably have antibodies in your nasal canal and in your lungs, right? And so that's where, you know, just for the sake of convenience, we may be measuring something that's non-mechanistic because, you know, having antibodies in your blood doesn't necessarily mean you're going to have a respiratory infection prevented, right? But it could be useful for us, right? Because again, we don't want to have to do sort of invasive measurements on people to understand what's happening in their mucosa. So this is a silly example, but I think a pretty important one to distinguish between how we think about different classes of correlates of protection. And there's not to say necessarily that one is better than the other. It sort of depends on the use case, right? As I just described, right? If you want to use an endpoint, a surrogate endpoint that's antibodies in your blood, that's a lot more convenient than having to do invasive measurements, you know, of what's going on in your mucosa. So yeah, so I mean, I think in general, we would prefer our correlates to be mechanistic. But you know, we can also sort of make progress on a lot of those use cases for correlates without ever claiming that we have a mechanism in our hands. Okay, so there's also a distinction between that's made in this field between what's called an absolute correlative protection versus a relative correlative protection. You'll see that I've started to abbreviate correlative protection as COP, which is commonly done in this field. So if I say COP, please know that's what I'm referring to. So absolute versus relative correlative protection. What's shown on the left is sort of the dream, I think. We really wish that correlates of protection worked like this. So the idea there is we've identified some immune response, right? Such that if you have an immune response below a particular level, so if you're somewhere kind of over here on this curve, the vaccine is doing nothing for you, right? If the vaccine isn't generating an immune response at least this high, you're not protected at all. But then once you cross this magical threshold, when you're on the other side of it, the vaccine is 100% effective. And wouldn't it be nice if mechanistically correlates worked like this? If so, we would call it an absolute correlative protection, and that would be very convenient and useful for us. The more realistic setting is probably more one like on the right here, where, you know, unfortunately, nature doesn't always behave in thresholds. There's something a bit more smooth and continuous, right? Where some immune response here is good. If you have low level immune response, maybe the vaccine provides some protection, right? But as you increase that, it's sort of a smoother transition to higher effectiveness. If that's the case, we say that we have a relative COP. And I guess I would claim that almost always in the examples that I've seen, we kind of live on that right side of the world, right? Is that if you have some antibody response to a vaccine, for example, that's good. If you have more, it's generally better. And eventually that curve sort of flattens off such that, you know, above some threshold, really adding more antibodies on top is not doing a whole lot. But in an ideal world, right, I think, you know, some people like to think in terms of absolute COPs. Certainly regulators wish they could think in terms of absolute COPs because it makes designing sort of endpoints for these like immunogenicity studies, like your seasonal flu vaccine studies that are very easy. You can just check in that trial, right, what percentage of people who you vaccinate get a response above that threshold. That is now exactly VE, right? So you can get a direct read off of VE just by looking at something very simple, like a binary endpoint, the percent of people who are above that magic threshold. More generally, as I said, things behave like relative COPs, so the regulators have to use their brains a little bit more to think about how to define endpoints in the context of these accelerated approvals that are a little bit more granular. So I think typically for flu vaccines, they have to show, you know, some kind of percentage above a threshold. I think it's like, what is it, like 4-fold increase in titers, I think is what they need for flu vaccines, but you also have to show something about the geometric mean titer. So it's not just about the percentage you're above the threshold, but also at what level are they above that threshold, right? And that's kind of reflecting that if you see both of those things, like a mean immune response that's pretty high, and a percentage above a particular threshold, that overall the vaccine efficacy is going to look pretty good. Okay, so moving on a bit, Cheryl, this is where I'm looking at you. Tell me where I get crazy, because I'm going to dip my toes in something I barely understand here, right? So I want to make the big picture point, irrespective of the bad biology I'm about to try to instill on you, that correlates of protection are not, you know, so far I've just been calling them immune responses. Really, these are readouts from assays. And the assay that you use matters a lot here. It's operating characteristics, the technical variability, right? How much day-to-day the assay readouts may vary, how much they vary from lab to lab. All of these things are, of course, critically important when you're trying to make decisions based on these readouts, right? So it pays to understand at least a little bit about the assays that are used to measure these immune responses, because it's nice to say like antibodies are the correlate, right? But that's not always specific enough because there's lots of different ways to measure antibodies and the function that those antibodies have. So I think broadly we can think about kind of two categories of immune response. They're the ones that I showed way back here, right? So there's this sort of humoral immunity. So this is like B cell antibodies. This is antibodies that are making. Okay, and the other one is this one up here. It's what your T cells are doing. Call that cellular immunity. Okay, so those are the sort of broad classes of immune responses that we can group things into. So antibodies versus cellular, and within each class, there's a lot of different assays that you can use to measure different things about what your antibodies are doing, how many of them there are, what their functions are, and ditto for cellular immunity as well. So as I said, mostly I would say correlates and protection focuses on antibodies for reasons that I'll mention in just a second. So I'll start there and talk about just kind of a distinction between two different kinds of assays. So binding antibody assays and then neutralizing assays. So it's either pseudovirus or live virus neutralizing. Okay, so here's a picture of basically what a binding antibody assay does. So you have a little well on a plate, and you have some viral particles that you've stuck to the bottom of that well, some viral proteins. So like for SARS-CoV-2, these would be like spike proteins. Right, and into that well, you're going to place some vaccinated individual's serum. So you took a blood draw, right? You isolated the serum from that blood, right? And now you're putting a little bit of serum on top of that. So if there are antibodies in that person's blood, right? What do antibodies do? They're trained to stick to the little spike proteins, right? So if you have antibodies in your blood, they fall down to the bottom of the well, they stick to those spike proteins, they bind to those spike proteins, right? And then we add this sort of enzyme labeled antibody. You add a little enzyme on top of it that makes the little well glow a certain color. And you can read what color that is glowing, and that tells you how much antibody there is in the blood. How'd I do, Cheryl? Good. Okay, all right, yes. Okay, so what is this actually measuring? It's measuring how well your antibodies stick to stuff, right? It's measuring how well they stick to a particular protein. And as I said, that's one of the things that your antibodies are really good at. They're good at sticking to things. That's like one of their whole purposes, is just to grab onto things and hang out there. We think that if you stick to a bunch of spike proteins, for example, on SARS-CoV-2, those little spike proteins are the same thing that the virus uses to grab onto your cell, right? And as long as you have something sticking there, maybe the virus can no longer grab onto your cell. So we think if we have really sticky antibodies, right, then these might be effective at preventing your cells from becoming infected and ultimately preventing you from becoming sick. But with this assay alone, we don't necessarily know that because we're just measuring how sticky things are. We're not measuring that whole sort of process of how these viruses go about infecting your cells. So for that, we have other assays. So we have neutralization assays that look similar. So here, you know, what we're going to do is, again, have sort of a little well in which we have stuck some pseudoviruses. So we've made a virus that kind of looks like SARS-CoV-2. It has the same sort of spike proteins, at least on its envelope. Right, and we've messed with that virus a little bit so that it glows a particular color at the end of the day, but for the most part, we've made a fake virus that has spike proteins sticking out of it. Right, and now we're going to do the same thing. We're going to add some vaccinated individual sera on top of those, and now we're going to measure how well that those little pseudoviruses are able to infect human cells. So we've sort of messed with these human cells a little bit to make sure that they glow the right color so that we can tell at the end of the day whether they've been infected or not. So the idea here is you're getting closer to measuring the function of the antibody because as part of this assay, right, you have this whole virus, you're trying to let it grab onto the cells, let it infect them, and you're learning whether your antibodies have stuck to it enough to stop that binding from happening, to stop your cells from becoming infected. So we can do this with pseudoviruses that we make that are, you know, harmless. They just have spike proteins on them. You can also do this with live viruses. So rather than having the fake pseudovirus, we have an actual SARS-CoV-2 virus that's there. Right, and we sort of can see whether that live virus is infecting your cells, right? Which is really the closest thing maybe to the actual biology that's happening in your cells, right? But it's also comes with the risk of having live viruses floating around in the lab, right? So not every lab is set up to be able to have live virus neutralizing assays to be able to run there. You have to have a certain level of biosafety to be approved to do those type of things. Okay, so what's the takeaway here is that, you know, assays, it's not good enough just to say that we're measuring antibodies, right? We have to be very specific about how we're measuring them, and in particular, how functional the assays are that we're measuring them with. Because we think that something like a neutralization assay is probably going to be a better candidate for a mechanistic correlative protection, right? Because it's sort of more realistically mimicking the mechanism that's happening inside your body. right? But they are more expensive, they're more time consuming, they require a lot more work, they pose greater biosafety hazards for live virus neutralization than binding assays, and so there's always going to be trade-offs here, right? Again, there's no one right or wrong assay to use, it's going to depend on the particular use case, right? And again, just the point is that, again, even if we have a functional assay, that may not be mechanistic, because again, you could, depending on the endpoint you care about, it could be a question of, how well is neutralization happening in your bloodstream? Is that really mechanistic for example, whether or not you get a respiratory infection? Okay, beyond this, these sort of two assays, there's a lot of different antibody assays that are available now that can measure a lot of different functions of antibodies. So antibodies do a lot of things that I barely understand. Like one thing that they do is sometimes they latch onto things and then they recruit other cells to come and eat the thing that they latched onto. We call that phagocytosis. And so we have things like ADCP, and we have assays that can measure that. So it's how well do these antibodies stick, and then recruit other cells to come and eat them and neutralize the virus in that way. There's now like sort of a whole field, I would say, of correlates of protection devoted to this. That paper that I'm citing there is a review study of recent development. We call this system serology. So these are like assays that are developed, they're really high throughput. And so they're generating, not just one antibody readout, right? They're generating like 120 or 200 different antibody readouts that are assessing different functions of the antibodies, right? And they're trying to identify correlates in that way. There's also cellular immunity assays. I think in the name of time, I'm not going to say a whole lot about these, other than sort of what my point is at the bottom here, right? Is that assays for cellular immunity require slightly different methods for sample collection and in particular storage. So whereas before we can measure humoral immunity by taking a blood draw and isolating serum from it, right? In order to measure cellular immunity, we actually have to separate the cells out of your blood, right? And be able to store those in a freezer. I understand this is quite challenging and quite expensive to be able to do. And so as a result, there's actually pretty limited capacity, I would say, to do this at large scale. So like, for example, as much money as we dumped into the COVID vaccine development pipeline in the US during the pandemic, right? We did not set up our clinical trials to collect the right specimens on people to be able to assess cellular immunity in those large phase 3 studies, right? The capacity just wasn't there in labs across our country. And so there are a few, trials that and trial networks that have invested very heavily in cellular immunity capacity to measure these things at scale. So the HIV Vaccine Trials Network did a lot of this because it's thought that HIV vaccines may have a mechanism that relies heavily on cellular immunity, but this is still a little bit, I would say, novel to be able to see studies of of cellular immunity at very, very large scales. So I'm going to just maybe push those aside and say that they're out there, but for the most part, most everybody in this field is focusing on antibodies. Is that fair?
That's fair. Yeah. Okay. It's expensive for, but a lot of trials might do some sort of immunology sub cohorts. You might get 10,000 people, and then you have like 1,000 or something that you might get those cells.
Right. Yeah. Right. And depending on the disease, maybe that's good enough to start to establish correlates, but for the most part, it might not be. So we'll talk about the designs in just a second. Okay, so getting a little bit closer to, so we've learned about how we measure these things, all the cool things we can do with them. What kind of studies do we actually do to try to identify these correlates of protection for vaccines in practice? So, again, I don't work a lot in animal studies, but this is a critical area for identifying correlates of protection because we can sort of control infections. We can do challenge studies in animals a lot easier than in humans, and we can measure immune responses by sacrificing animals in ways that you can't with humans. And so animal studies are really critical for helping establish vaccine correlates of protection. And so just to give you a flavor of what some of these studies can look like. So this is from, again, another early COVID vaccine development trial that happened in non-human primates, right? So these primates are given a vaccine, and after a certain waiting period are challenged with SARS-CoV-2. So they have to inhale, SARS-CoV-2 viruses into their airways. And then eventually the animal is sacrificed, and we can check and see things like how much virus made it to their lungs, how much virus is in their upper airways, right? And so we also have on that animal at the day that they were challenged, we take a blood draw and we can measure one of these immune responses with one of these assays we just talked about. So here's like an RBD specific IgG, that's like a binding assay. And so what you see going on the bottom horizontal axis there, right, is the level of antibody activity. So going further this way means that the monkey had higher antibody levels. Going on the vertical axis is RNA copies. So that's basically how much virus was replicating. I don't remember if it was the upper or lower respiratory tract or something like that. And so you can see that there's a negative correlation here, right? The more antibody the monkey had, the less viral replication there was, supporting the hypothesis that that RBD-specific IgG could be a correlate of protection. Okay, so we see that, the more antibody you had, right, the less the virus was able to replicate, providing some evidence that antibodies are playing a role here in protection. Another thing you can do is passive transfer studies. So this can be done in animals, it can be done in humans, but basically you have a vaccinated or naturally infected animal, right, that generates an immune response. You can isolate the antibodies from that animal and passively transfer them to another animal, and then challenge that animal with SARS-CoV-2, right? And if you see that animal is protected through that passive transfer of antibodies, again, that's providing some evidence that antibodies are mechanistically playing a role in protection. So I don't have a lot more to say about animal studies because, again, it's not really my cup of tea, so I think I'll move on, but if there's questions at the end, we can come back to that. Where I work a little bit more is in phase 3 trials. So using phase 3 trials, randomized trials of vaccines to generate evidence in support of correlates of protection. So I'm just giving a schematic here for a kind of standard design of a phase 3 randomized trial. And in particular, this was for the AstraZeneca COVID vaccine. So there's kind of 2-to-1 randomization to the AstraZeneca vaccine product or a placebo. So you can see sort of study visit days that are going on that horizontal axis there. So they came in at day 1, they got their first dose of vaccine. They came in at day 8, they probably filled out some paperwork to, say what kind of reactions they had to the vaccine. They came in at day 15 and we took a blood draw. We would use that blood draw potentially to measure immune responses. They came in at day 29, they got a second dose of the vaccine a couple of weeks later. We took another blood draw there at day 43 that we could potentially use to measure immune responses. And then we follow those individuals up over time, right? Some of them get COVID, some of them get severe COVID, some of them get asymptomatic COVID infections, right? And those are the data that we then have to say, okay, can we make some kind of claim about in humans, right? That we have some kind of antibody assay that's acting as a correlate of protection. So, I'm a dorky statistician, so I had to say one thing about dorky statistic things. So what's kind of interesting about these trials is that, generally, we are encouraging serum collection on all trial participants. So when we design these trials at day 15 and day 43, everyone in the trial is going to have a blood draw. Okay, but we're not going to measure, because this trial had 30,000 people enrolled in it, we're not going to run an assay on 60,000 samples. That would take a very, very long time, and it would be very, very expensive. So instead, what we do is we take this collection on all trial participants, and we store it in a freezer, right, until the trial is over, until some point into the trial, where we implement a particular sampling design to try to make this a little bit more efficient. So we say basically, who are the most interesting people in this trial to learn about their immune responses? The most interesting people are the vaccinated individuals who, in spite of getting vaccine, ended up having COVID. Okay, because we want to know what happened to them, right? Did the vaccine fail, right? Does their immune response somehow look a little bit wonky? And just from a statistical perspective, we have all the power, that we're deriving in a study is from the actual endpoints that we observe. Okay, so controls are not really giving us too much in terms of power. So the most interesting people are the people who end up having the COVID endpoint. So we go back to the freezer and we pull all of the samples for people who end up getting COVID, and we measure their immune responses from those samples. Right, and then we augment that with a randomly selected subcohort. So we just randomly pick people from the cohort. And we go and pull their samples from the freezer and we measure them as well. We call this design case cohort sampling. And it turns out that statistically, it's almost as efficient as if you had measured immune responses on every single person in the trial. So it's a pretty neat design that allows us to move very quickly, right? Be very efficient with our resources and still at the end of the day, learn what we need to about correlates of protection. All right, so I've you're lucky, I've run out of time to walk through this very dense slide where we talk about all the cool different analyses that we can do of those data once we've generated it, and all of the subtle ways that we can ask kind of causally related questions about how correlates are behaving. If that's your cup of tea, I'll just point you to this paper that's referenced at the bottom. There's lots of cool analyses that you can do of these to try to infer different things about whether or not, particular assay readout is a good candidate for being a COP or not. Okay, so maybe just with the last few minutes here, so I'll save a few minutes for questions. I'll show you just a few of what these results kind of look like after you analyze the data? What kind of evidence, what kind of things would you see in that data analysis that would support something being a correlative protection? Okay, so the first one is like a very sort of classic thing that's on the left here. So in this trial, we did this case cohort design. We did all the fancy statistics to account for that design. And what we're showing over there is basically a box plot that is showing the distribution of, in this case, neutralizing antibody titers. This was a dengue vaccine trial, right? And what we're showing is that what we see in the vaccine control, so these are the people that did not get dengue, right? Those are the people that tended to have a higher antibody response, right? The people who did get dengue when they got the vaccine, they tended to have a lower antibody response, right? So we see this. This is evidence, this separation of these distributions is evidence that maybe this neutralizing antibody titer is functioning as a correlative protection, right? If you have more antibody level, right, you're more likely to be a control than a case. You can also show this in terms of incidence. So just doing a simple analysis here where you show like the cumulative incidence by neutralizing antibody titer level. So you can like break this into tertiles, right? So the lowest tertile of antibody responses here has the highest incidence. The middle tertile of antibody responses has kind of been the middle incidence, and the highest antibody tertile has the lowest incidence, again, providing some evidence of this neutralizing antibody titer functioning as a correlative protection. You may have noticed that you also see some differentiation here in placebo, right? So you have a similar thing, and this is because this was a study of dengue, and there were kids who had previously been infected with dengue, right? So even in the placebo arm, you can see these neutralizing antibody titers. So even in the context of getting a placebo, if you had high neutralizing antibody titers at baseline, you had low incidence, indicating again that probably antibodies have something to do with protecting you from getting future dengue infection. Another thing you can do here is make a plot like this one on the right that tries to say this is really a method that a friend of mine developed that really tries to hope that there's some kind of absolute correlative protection. So there's some threshold above which you start to get protection. That's what this method is trying to do. And it's basically trying to say if you have immune responses at least, let's say a titer of around 100, right, that we can expect that you have about 25% vaccine efficacy. But as that titer increases, you know, you'll go up to about 100% there. So again, that's this sort of idea that some is good, more is better. We can kind of break this into absolute versus relative COPs, generate data in that way. So here's those same kind of curves for COVID-19 vaccines across a number of trials that we studied that were funded by the US government. Again, kind of showing how, again, the horizontal axis here is an antibody titer, an antibody readout, more higher values on this side, right, are indicating you have better antibody responses. And on the vertical axis, we're showing what the estimated vaccine efficacy is for someone who has that antibody titer. So again, evidence for COVID-19 vaccines that neutralizing antibodies were functioning as a correlative protection. I think I'll skip that. And then finally just say that some people, you know, go even further than this and start to meta-analyze these things. And that's where you're really generating robust evidence that you have the correlative protection is if you're seeing the same association time and time again, where if you have higher antibody levels, right, you're doing a good job of predicting whether or not a vaccine works. So this group out of Australia, led by Miles Davenport, did this a lot for COVID-19 vaccines, where we saw at least early on, it was pretty incredible how accurately you could predict clinical vaccine efficacy based on just neutralizing antibody titer readout. So you can see plots like this, where the vertical axis or the horizontal axis is again here, this is neutralization level. And on the vertical axis is vaccine efficacy. And they're showing this for all sorts of different vaccines that were studied. So Pfizer, Novavax, Moderna's up there, the Johnson & Johnson vaccine, the Oxford AstraZeneca vaccine, CoronaVac, all of them you could predict pretty much dead accurately just by studying neutralizing antibody titer. Okay, so in conclusion, vaccine correlates of protection are pretty cool. Something that we should probably continue to invest federal dollars funding research in, right? Right. So myriad uses for correlates of protection across the vaccine development pipeline. And again, just trying to make the point through this talk that really, I think COPS are an example where, you know, the types of training that this program is getting, appreciating how infectious disease functions across scales from basic biology up to human behavior is pretty important. Final point is, you know, this is a weird time. I don't have any sage words of wisdom other than, you know, I hope that within programs like this and within communities like this, throughout this, we can keep supporting each other and keep supporting science and keep doing the awesome work that we're doing. So appreciate the time. Thanks for having me out, and happy to stick around if there's questions.
Questions on the chat?
Not yet.
Yeah, please.
Yeah, I thought it was a great talk. Thank you so much. I have kind of two questions, both about how correlates of protection vary or if they vary. The first is kind of maybe at like a smaller scale. Would you say that is it fair to say that correlates of protection can vary depending on like the way that you become infected? Like I'm thinking about HIV, for example, where you get sexual transmission, like that's generally kind of the family viral population is generally much smaller than if you're like injecting or something like that. Does that have any impact on what correlative protection? And then maybe like related to that, does it matter? Okay, next question, sorry.
Sure. Yes, I think is my answer. I think it's like when we say, can correlative protection vary by like mode of transmission or something? I think like the answer is yes, but we have to like sort of specify like what are we trying to use a correlative protector, like to what end are we trying to use it? And for that end is important to kind of distinguish between these and and how would that distinction then influence how we would design a study, let's say, to try to do these. Like so, for example, like if you did like an NHP challenge trial or something like that, like you could see very different, like if you like rectally challenged monkeys with HIV versus challenge them with an injection or something, you could identify very different correlates, I think, in that setting, sure. I think in human trials, I don't know. Yeah, I'm struggling to think of a situation where that distinction would be relevant for like kind of at that scale, but certainly, for vaccine development and biological understanding, I could see, yeah, definitely mechanism, mode of transmission could really impact what the correlate is, yeah.
Okay, and then, thank you. And then my second question is, related to a little bit like the flu vaccine that you were, or the example of flu that you were talking about. So for something like flu in particular, I think like obviously one of the reasons that you vaccinate people is because you hope that they don't get sick. But another reason that you vaccinate people is because you hope that they don't get other people sick.
Yeah.
Which is related to like mucosal immunity versus like immunity in the bloodstream. And yeah, so I guess my question is like, are these like clinical trials, the data that you see, are they generally kind of only assessing correlative protection as it relates to preventing infection in individuals? Or is there ever data that's gathered in terms of for correlative protection against transmission? Yeah, does that like exist or is it being done or not so much?
It's hard to do,
Yeah.
right? The transmission studies. Yes, it's being done. I think for COVID, there's a big project I'm aware of that's funded to look at transmission blocking vaccines, so mucosal vaccines, inhaled vaccines, and correlates is an element of that. I think the plan is to go with human challenge trials for that because you kind of need that level of control to be able to measure how much shedding is happening from these people. And you hope that the shedding that you measure is going to be like surrogate for how they would transmit to household members or something. Yeah. I'm not sure. I mean, surely there are probably household transmission studies that have collected immune responses. I don't see a lot of that out there. But yeah, you're absolutely right that, it's a critical distinction. The correlative protection is defined not just by like the assay you're using to measure it, but also with respect to the endpoint. It correlates with vaccine efficacy for what?
Right.
Preventing infection or disease or severe disease, right? And those could all be very different mechanistically, in terms of what immune responses matter most. And so we try to do that to the extent possible. Like when we do the COVID trials, we'll report on correlates for asymptomatic infections because we do serology over time. So we know people when they have asymptomatic infections. Severe disease is harder because we don't see that many cases, but we'll try it and see what evidence we can generate. But yeah, it's very often a data limitation that you can't study every endpoint that you might like.
We've got a question online. Anya, would you want to unmute yourself and ask your question?
Yes, sure. I had a question on the dengue vaccine analysis that you showed. Yes, the next, yes. So in the left-hand side, the way I understand this graph is basically people who are vaccinated and never got the infection probably has the highest level of antibody titer. And basically, my question was that what is happening in the vaccinated people who got infection versus the placebo people who didn't get the infection. So I I feel like they have similar levels of antibody. So then what would your conclusion from that be?
So your observation is that the placebo control, so people got placebo and were not infected, have similar antibody levels to the vaccine cases.
Yes. So does that mean
Yeah,
antibody level is not like protective or whatever? I don't know.
yeah, I think it's a really important question. I mean, so it's been years since I looked at this dengue case. I'd have to remind myself about the vaccine construct, but an important thing to remember is that natural infection will stimulate different immune responses than infection or what controlled infection with a vaccine. Because generally, you're being exposed to many different antigens, the whole envelope protein. So you maybe have an antibody response, but you also have cellular immunity, and maybe your antibody response is a little bit different than what would have been generated in response to vaccination. So, in these contexts, I think it becomes important, but somewhat difficult to start to compare across the placebo controls whose antibodies are generated in response to a natural infection versus the vaccine cases whose antibodies are not necessarily, those may be only vaccine infused. I think in both cases here, both the placebo controls versus placebo cases and vaccine controls versus vaccine cases point to sort of antibody neutralization as likely being important for protection, but not telling the entire story. Because, you don't have complete separation here, right? Like what you might have if you had a really great top, right, is that all the vaccine controls would be up there by 10 to the 5, and all the vaccine cases would be down there at like 10, right? And so again, this is sort of saying, yeah, we have something that maybe we can base some decision making on, but it's probably not telling the whole immunological story, whether that's in the context of vaccination or in the context of natural infection.
Yeah, thank you.
My question was along the similar lines of Ananya. I'm curious about things like antibody dependent enhancement in dengue, and where like a lot of antibody seems to be fairly protective, but then a little bit of antibody might be enhancing. How do you deal with that in terms of protection? Do you just kind of choose a time?
Yeah, I got myself in trouble by showing the dengue example, right? Because that's the one where like it is really complicated, and I'm not a dengue person, and I haven't kept up with that field. Yeah. So I would say when this analysis was done, this was like prior to the observation that there may be, and this is the CYD 14-15 vaccine. So this is the one that there is a concern about antibody dependent enhancement. But this was published before that was really kind of noted as a concern. So I'm actually not sure about kind of the state of things with that. But I don't have a great answer. Yeah. Sure. Yeah, sure.
So, yeah, my question, or just the difficulty interpreting some of these data, or like what your definition is to be able to call something a correlative protection, because you can get, if you have a lot of data points and a lot of power, you can have a small difference and claim that it's in a p-value, and if you have a p-value, then people say, oh, this is a correlate. But as you see this type of data, you have almost like a huge overlap in people are protected, not protected. So I don't know if there's really a right answer to that, but just like how good does that data or have to be be able to call a correlate as if you get your p-value less than 0.05? I'm just putting out there that like that doesn't necessarily mean that biologically that readout wherever it's always going to be protective, because you do end up having overlap in the level, like antibody levels to equally overlap, but when you do several hundred people and you have your medians here and here, you say, oh, see, it's a difference, and there's generally more. But I think it's just, as you're sort of pointed out already, that it could be nice to have this like black and white.
Yep.
But it's like, that's not really, mostly that's not real life either. So this is a challenge, I think, the fields are really, how confident, how good does that have to be for us to confidently say this is a true correlate?
Yeah, no, and I probably should have added in the slide, usually when I get talked to this, there is a slide that like, there's probably no one analysis that's ever going to constitute enough evidence to claim that something is a correlate. And as you say, like, even if you have statistical evidence, that might not make it sort of clinically meaningful to make decision making on. But again, it kind of depends. Like,
Depends on the.
For like lot to lot consistency, probably who cares, right? Like, that's probably good enough. But like to make accelerated approvals, you need a much higher burden of it. So I think like, it's all about triangulating all of the data sources, you know, from animal models to the human studies. If you want to talk about antibodies, you know, with like COVID, for example, we also had monoclonal studies that showed prevention, you know, through monoclonal fusion. And so that's saying that like antibodies, you know, are doing something mechanistically here.
Yeah, for that, and like how good, what is your threshold for vaccine efficacy? Like, are we happy with the vaccine efficacy of 50% or 30%? Again,
Yeah.
what is the disease? What's the prevalence in the population? 30% might actually be beneficial versus some cases you think they're
Totally.
a horrible vaccine efficacy.
Totally, yeah, it can't be black and white, right, in terms of decision making. You have to have evidence coming from all quarters, and it has to always be put in like the right epidemiologic context to say like, is this appropriate public health decision making based on the current state of knowledge? Yeah. Great.
I think we've come to an end. Thank you.